Hi there. Today we're going to learn all about gene editing together. We're going to talk about gene editing technology in the future, and we're going to talk about how incredible advances in the history of cancer treatment have been in the use of one particular technology, CRISPR, and we're going to talk about ethical issues to think about and what kind of orientation we need to take in the future. Have you ever watched these two movies? If you have, what do you think they have in common? Yes, that's right. The main thing they had in common was the technology of gene editing. Well, when you think of gene editing, you can think of a lot of keywords. When I first heard it, I wanted to learn more about the actual use of that technology, the various related biotechnology, and some history involved. And basically, that's why my group came up with the presentation topic relatively easily. The first thing I started with was searching the internet. I learned about the definition of gene editing as a technology that allows scientists to make specific changes to an organization's DNA, so much as adding, deleting, or alternating specific sequences. And in the course of further searching, I learned that there was this special interesting technology called CRISPR. CRISPR technology is truly helpful treatment and it is a key to the enhanced health of humanity. But it didn't come up out of nowhere. Before that, there was TALEN existed. TALEN emerged as a second-generation gene editing tool around 2009, significantly improving upon earlier ZFN, which refers to zinc finger nuclease, a tool used in genome editing to make precise cuts in DNA at a specific location. Anyway, back to TALEN, its core principle relies on Tal effector proteins. You'll probably haven't heard this thing too. Let's imagine Tal effector proteins are like the super smart GPS for our gene editing tool, TALEN. Each little part of these proteins knows exactly how to read a specific DNA letter, so we can basically custom build them to find almost any DNA sequence we want. When two of these customized proteins successfully grab onto their target spots on the DNA, they bring a special cutter enzyme right together to make a precise break. Pretty neat how they guide the whole thing, right? But TALEN still faced numerous limitations, including its protein-based nature, which made its delivery into cells pretty challenging, and its synthesis process more complex than the subsequent CRISPR system. Finally, here comes the CRISPR, also called Cas9. CRISPR simply edits genes by precisely cutting DNA, and then harnessing natural DNA repair processes to modify the gene in the desired manner. The system has two components, the Cas9 enzyme, and a guide RNA, also called as gRNA. Initially, we primarily relied on surgery to physically remove tumors, which was pretty much our only option for centuries. Then came radiation therapy, using focused energy to destroy cancer cells and shrink tumors from the outside. Next up, chemotherapy emerged, a systemic approach using powerful drugs to kill rapidly dividing cells throughout the entire body. More recently, targeted therapy arrived, a game changer that specifically attacks cancer cells by focusing on their unique molecular vulnerabilities, leaving healthy cells mostly alone. Now, we're really getting futuristic with immunotherapy and gene therapy, which harness the body's own immune system or directly modify genes to fight cancer from within. Okay, so while gene editing is super exciting, it opens up a huge ethical can of worms. One big worry is the concept of designer babies, where parents might choose traits beyond just health, fundamentally changing what it means to be human. Then there's the critical safety issue of unpredictable off-target effects and other safety risks, meaning we might accidentally edit genes we didn't intend to, with unknown and potentially harmful consequences. Lastly, there's a serious concern about the exacerbation of social and genetic inequality. If only the wealthy can afford these enhancements, it could create a deeper divide between the genetically enhanced and everyone else. Given these complex challenges, the very first attitude we absolutely must embrace is a profound sense of responsibility. This means rigorously ensuring safety, fair access, and constantly discussing the societal impact as we move forward. Believe it or not, they basically pulled dinosaur DNA from ancient mosquitoes trapped in amber, and then, for the missing pieces, they just spliced in frog DNA. Yes, frog DNA. That's why their dinosaurs can sometimes change sex and totally wreak havoc. Who knew amphibian genetics were so powerful, right? It's genius storytelling, but scientifically, that's where the not part of believe it or not kicks in pretty hard. Mammoth Revival Project happened in similar course, too. If you got interested, you can go search for it right now. Thank you for following the long presentation well. Bye bye.